Hello everyone! So today we're going to talk about the effect of temperature on enzyme activity. But before that, for those who doesn't know me, I'm salsabila khairunnisa from Semarang State University. And now let's go to the topic. There are 2 points that we'll get highlight today. The first one is enzyme and the second one is temperature. So, what exactly is the relationship between these 2 points? And what is enzyme? Enzymes are biomolecules that function as a catalyst in a chemical reaction. The catalyst itself means a compound that can change or speed up the rate of reaction without being involved into the reaction. There are various factors that affect enzyme activity, namely temperature, pH, substrate concentration, enzyme concentration, and the presence of inhibitors. At very low temperatures, enzyme activity can be reversibly stopped. An increase in ambient temperature will increase the kinetic energy of enzyme and the frequency of collisions between the enzyme molecule and the substrate, so that the enzyme becomes active. The objective of this topic, of course, to determine the effect of temperature on enzyme activity. Now, onto the methods. First, we have to prepare 2% starch solution and amylase enzyme. Then, give each tube 2 ml of 2% starch solution plus 1 ml of amylase enzyme. Then, give a different treatment to each test tube that has been filled. Tube 1 is inserted into a beaker glass filled with ice water. Tube 2 was left at room temperature. Tube 3 is put into a water bath with a temperature of 37 to 40 degrees Celsius. Tube 4 is put into a water bath with a temperature of 75 to 80 degrees Celsius. And the last tube is placed into a boiling water bath. Then leave it for 15 minutes and test it with iodine and Benedict's reagent. Next, there is the results. The test tube that shows a positive results for iodine test is the tube 1 with cold water, the tube 4 with 75 to 80 degrees Celsius, and tube 5 with boiling water. And the test tube that shows a positive results for Benedict test is tube 2 with room temperature and tube 3 with 37 to 40 degrees Celsius. A positive results for iodine test is indicated by the color of the solutions turning into blackish blue or purple. Meanwhile, the Benedict test is turning into brick red precipitate. And the function of iodine test itself is to detect the presence of starch, and the Benedict to detect the presence of monosaccharides. Then in here, we can see the reaction process from Benedict and iodine test. When starch meets iodine reagent, it will produce a blackish or purple color. And when the monosaccharides meets Benedict reagent, it will produce a brick red precipitate. When the amylase enzyme can work optimally, it will break down starch into monosaccharides. Meanwhile, if the amylase enzyme cannot work optimally, that means inactive or denaturated, then it cannot break down starch into monosaccharides. From all of that, we know the enzyme in tube 1 is inactive. That means it still contains starch, because the starch is not break down into monosaccharides. And for the tube 2, the enzyme is active. That means the starch is break down into monosaccharides, and same for the tube 3. And for the tube 4 and 5, the enzymes is denaturated. That means the starch is not break down into monosaccharides, then the starch is still there. So the conclusion of this topic is temperature is one of the factors that affect enzyme activity. In general, enzymes can work optimally at a temperature of 37 to 40 degrees Celsius. If the temperature is too low or under 20 degrees Celsius, then the enzyme will be inactive. And also, if the temperature is too high or 50 to 100 degrees Celsius, then the enzyme will be denatured. So, that's all for today. Thank you so much for your attention. See you in the next video. Bye.